Hi guys, nice to see everybody Tuesday afternoon. Therefore, a little bit of a biology carol lesson. And as I think you are already expecting, we're going to be going through AQA Biology Paper 2. Now, my big challenge on this is to have some idea as to how slowly or how fast to go through this, because I don't know how much time to give you to read the questions, whether you'd like me to just go through it fairly quickly. So actually comments in the chat would be really useful as to whether I need to give you more time to read the questions, whether my kind of synopsis of the question is enough. Just a little bit of feedback on that would be really helpful, so that because I don't want to feel like I'm going through this at a snail's pace, because I think hopefully if I can get the pace about right, this might be useful and we might be able to get through a reasonable amount of the paper. So what I've done for each one, I'm going to write out my the answer that I gave. I'm going to show you the mark scheme and I'm going to show you the marks that I got for the exam. So this is all about heat stress, photosynthesis, and it tells you that heat stress decreases the light-dependent reaction for photosynthesis. And you've got to explain why this leads to a decrease in the light-independent reaction. And that, of course, hopefully, here's me giving the examiner abbreviations as to what I want, so that I'm not writing it all out in longhand. And that's because the light-dependent reaction will produce less ATP and less reduced NADP, which are both required for the light-independent reaction. And as a result, if there's less of them, there's going to be less of the light-independent reaction happening. Here's the mark scheme, and therefore I got both of the marks. Just worth you knowing that when I mark exam papers, I literally mark at the word, or I try and mark at the word where the particular mark point has been earned. So hence, mark point 1, mark point 2 there. Hello, lovely to see you all. Just a bit of time to read the questions, maybe 30 seconds. The screen's a little bit, it's blurry, isn't it? Oh, has that, Hazel, I'm hoping that it is now completely cleared up. I don't know whether that is because as I change screens, but let's see how that goes. So, and I'll try and do a synopsis of the question as well, Munib and others. So, another effect is that Rubisco, which you should know what enzyme that is, the activity of the enzyme is decreased. And we are asked to explain why a decrease in this enzyme would limit the rate of photosynthesis. So I'm thinking, okay, I know what Rubisco does. It combines CO2 with RUBP to produce 2GP. I'm literally visualizing the Calvin cycle in my head. So this is the answer that I wrote down, because less carbon dioxide will be able to combine with RUBP. Now, here was my question. I didn't know, I knew that they would want this. This bit I was pretty sure of. Up until here, I was thinking I've got a mark. But limit the rate of photosynthesis. What was going to be the bit of the Calvin cycle that got me the mark that proved that photosynthesis was being limited? So at this point, I summarized the whole lot because I wasn't sure. So therefore, less GP would be made, which would mean less TP and therefore less glucose. GP, glycerate phosphate, TP, triose phosphate, therefore reducing the rate of photosynthesis. Pardon the tongue twister. Here's the mark scheme. I got both of the marks. Clearly didn't have to write as much as I did, and actually the GP this mark point should have been here, so apologies for that. So hopefully that explains my thinking of that and why I wrote what I did. I ended up writing too much, Don't see how I could have avoided that. And here it asks you specifically where Rubisco is found. And of course, you are supposed to know that the light independent reaction, the Calvin cycle, happens in the stroma of the chloroplast. And therefore, that's where Rubisco is. Right, first application question. You get given a couple of graphs. One about the activity of Rubisco activase, and this enzyme activates Rubisco. So, and you are told that the scientists concluded that heat stress reduces the activity of Rubisco in plant leaves by affecting Rubisco activase. Evaluate their conclusion. Now, evaluate means pros and cons. You're looking for arguments against and you're looking for arguments for the conclusion being the correct one. So here am I reminding myself for and against, pluses and minuses. But here am I now going back. This is what you have to do for an evaluation question. And this is an AO3 question. This is your first really higher level question in the paper. Notice that the scientists concluded that heat stress reduces the activity in Rubisco in plant leaves. Kind of going, okay, whoopee-doo. Now go back to see what the experiment did. The experiment was looking at the effect of temperature on the activity of two enzymes which were isolated from the leaf cells of cotton plants. Now, the scientists have concluded that this happened in plant leaves. That means in a living plant and in all plants. Well, the flipping experiment, the enzymes were isolated and they only used cotton plants for the experiment. So I am noting this. I'm ready to critique this in my evaluation. But first of all, I'm going to look at the data. And I want to kind of look for pros and cons in the data. So here am I, again, telling the examiner, Rubisco activase, I'm going to use RA, and Rubisco is R. So here am I kind of going, okay, so Rubisco activase leads to Rubisco being activated. So I'm kind of going, okay, so between 0 and about, 35 degrees, Rubisco activase activity increases. And that certainly is also the case for Rubisco. However, above 35 degrees, Rubisco activase activity decreases, but Rubisco carries on going up. So here is my 4 from the data. My 4 is Rubisco activase increases 0 to 35 degrees, just as Rubisco activity does as well. But the against is Rubisco activase activity decreases above 35 degrees, whereas the Rubisco activity carries on increasing. Now, at that point, I'm kind of going, I don't think I've got much more to say about the data. Now I'm going to go to critiquing the experiment. If there was more that I could obviously say about the data, I'd do it. So, and here am I now kind of going, however, results are only for cotton, not all plants, as the scientists have said. And also the results, the experiment is in isolated enzymes, whereas the conclusion has been made for all plant leaves. So really useful to remember that when you are evaluating data, look for the obvious things that are pros that support the conclusion, look for obvious things in the data that don't support the conclusion. And when you've run out of things to say about the data, then I would go to critiquing the experiment. And classic things to look for is where the conclusion is more broad than the experimental results allow. And also things like, sample size really small, those kinds of things. And generally you'll have enough to put together at least a 3 or 4 mark answer. So as it is, I got mark 0.1 because Rubisco activity increases with temperature, and I got mark 0.2 because Rubisco activase activity decreases at high temperatures, allow any temperature above 25 degrees. Well, I read the graph, so, you know. However, look, here, the rest of the results are either about critiquing the data or noticing that no stats test was done. Now, I didn't go down that route, but I could have done. So I therefore got all 4 marks for that question. Okay, so here one. 2 types of gene mutation. Put a tick in the box next to the statement which describes incorrectly the effect of the mutation in an exon of a gene. Okay, so we're looking for the thing that isn't correct. And if I give you a few seconds to have a look, hopefully you'll realise that this is the incorrect one. Because an inversion isn't going to change the number of DNA bases. Inversion is where a piece of DNA literally kind of goes, flips around. It's not going to change the number of DNA bases. All right. Deletions do result in a frame shift, and addition does result in a frame shift, and a substitution may not result in a change to an encoded amino acid because of the degenerate nature of the DNA code. So got that one. Okay. Describe how alterations to a tumor suppressor gene can lead to the development of tumors. Now, this is about reading the question, because notice it's not just about mutations, it's about alterations. Anything that alters a tumor suppressor gene. So I'm afraid I am thinking about increasing the amount of methylation. I'm also thinking about mutations, but also because this question is about the development of a tumor, I'm already thinking, I am almost certain that one of the mark points will be about saying that a tumor comes about as a result of uncontrolled cell division. So here am I. So here is, this is my abbreviation for the examiner. So hypermethylation, hyper means too much methylation. And remember that methylation is a type of epigenetics. Methylation minuses. So too much methylation will greatly decrease the expression of tumor suppressor genes. And actually, we like tumor suppressor genes because healthy tumor suppressor genes reduce the mitosis. They reduce cell division. They suppress tumors, okay? So if we decrease the expression of a tumor suppressor gene, does it make sense that we are increasing the likelihood of uncontrolled cell division and therefore the development of a tumor? Hence the first sentence. But I've also kind of realized, alterations, something else. Okay, something else they want. I didn't quite know where all the marks were. I'm thinking that I've got 2, hypermethylation and uncontrolled cell division. I'm kind of thinking 2. So I'm thinking this is my 3rd mark point. Mutations in the tumor suppressor gene could also result in the production of a non-functioning tumor suppressor protein, which would also lead to increased cell division. Hopefully that makes sense. Methylation, yes, methylation. Methylation is adding a methyl group. You cover that, tang, in epigenetics. You talk about methylation and acetylation. Methylation adds a methyl group and it ends up decreasing how much transcription happens. So, and acetylation ends up increasing transcription. You'll have to watch my epigenetics video for that one. So here's the mark scheme. Here are the notes in the mark scheme. Notice I got my mark point here because I could use the term hypermethylation. Absolutely accurate. Absolutely accurate. I got mark point 3. Tumor suppressor genes aren't transcribed or expressed. Uncontrolled cell division, mark point 4. And blow me down, all I had to write was that there was a mutation. So clearly I wrote too much, but as an A-level student, I don't think I'd dare writing less, if I'm really honest. Okay, first bit of maths. Here we go. Don't know what you like with your maths. So here is my silly little diagram. This is me getting my head around the maths. So every cell division doubles the number of cells. In notice, one cell division equals 2 cells, and 2 cell divisions equal 4 cells. Notice that 3 cell divisions will equal 8 cells. Now, those numbers might give you an idea as to what you've got to do with the maths. We have got to work out the number of cell divisions and then take 2 to the power of that number of divisions. Go through it as well. And the other factor is that each cell division takes an hour. So my first logic was, okay, how many 8 hours are there in 4 weeks? Because that will tell me how many cell divisions have occurred. Work that out, and we got to 84 divisions in 4 weeks. Now, for each division, you are doubling the number. So you are doing 2 to the power of 84. Notice that 8 is 2 to the power of 3. 2 times 2 times 2. Yeah? 2 squared is 4. 2 to the power of 1 is 2. So to get the number of cells that you'd have after 4 weeks, you've got to do 2 to the power of 84, which is a frighteningly large number. And if you put it into your calculator, this is what you get. And you're told to write it in standard form. So 1.93 times 10 to the 25. Here's your marks point. Look, if you got the 84 cell divisions, that got you a mark. If you weren't sure what to do with it, at least you got 1 mark for that. But I got both the marks there. Oh, quick, just your reminder, my website, bargain basement, video revision resources, 50 quid for year 12 and year 13, £25 for year 12. This is to get access to them for the whole year. You can earn your money back by telling your friends about it, and there's a freebie, so do feel free to go and have a look at least. That enables me to carry on making these videos. It is literally the thing that enables that to happen, because each of these takes such a long time to prepare and film and edit and all the rest of it. So anyway, right, on to question 3. Yeast cells can respire aerobically and anaerobically. Okay, fair enough. Use this apparatus, and you're told that it was in a water bath and it reached a constant temperature, and then they left the apparatus for 1 hour. Okay, all right. Suggest one reason why it was important that the student left the apparatus for 1 hour after the culture had reached a constant temperature. That tells you the answer is not so that it can reach equilibrium, which would have been a classic, but they've told you it's already reached constant temperature. So here am I going back to the question and going, right, what else in that diagram gives me a hint as to what the answer could be? So I'm looking at the diagram. And the thing that struck me first was that, oh, okay, then that hour might allow all the oxygen to be absorbed by pyrogallol. That's what I put. So, and here am I emphasizing that. So that's what I went for. And this was the answer. Notice I got it for here, obviously this bit, but rightly, you could have also said that it allowed the yeast to either use up all the oxygen in the flask or to respire it, or fair enough. Okay, during the investigation, coloured liquid moved to the right. Let's be clear, it's this way. All right, explain why it moved to the right. This is a classic question. It comes up in various forms. An experiment to do with, potometers, respirometers, what else? Anything involving a movement of a flipping bubble or a coloured piece of liquid. Really common. Why does the bubble or liquid move? And I know that one of the mark points will be, oh, it's because the pressure in the flask increased. So now all I've got to do is think about what causes that pressure in the flask to increase. Well, I know from my biology that when the yeast, now notice we've already established that this is now anaerobic respiration because the apparatus has been left for an hour, all the oxygen has been used up, that this yeast is going to produce carbon dioxide. As a result, the volume of carbon dioxide in the flask, let me just find where my pointer is, there it is, okay, the volume in the flask will increase, the pressure will increase, and therefore it's going to force the bubble to move to the right. So, because yeast respiration, anaerobic. So notice I've written the word respiration, and I thought, hold on, I've just written the word respiration. Do I know whether it's aerobic or anaerobic? Yes, I do. I'm writing it in. Okay, just a little hint there. If you know the type of respiration, write it in, because at A level, that is often knowing that and showing that you know it is what gets you the mark. Will produce CO2, increase the pressure in the flask, causing the bubble to move. So here we go. You needed anaerobic respiration to get the mark. And this whole thing about increasing the pressure or the volume of the gas got you the second mark. So there we go. Now, this is another classic maths question where there is an experiment which uses a capillary tube. It's really common. Student found that the coloured liquids moved 1.5 centimetres in 2 hours. That ain't very much, but hey ho, whatever. Diameter of the lumen, the hole in the capillary tube, was 1 millimetre. Okay, volume of capillary tubing is given this. This gives you a huge hint, and you're told what all of the bits mean. Okay, so here is my capillary tube. I've drawn a very long, slim cylinder. I know the length is 1.5 centimetres and I know the diameter is 1 millimetre. So, and here's the crucial thing. If you don't know what you're doing, look at the units at the end of the question. They want you to work out centimetres cubed of something, and you probably know it's the gas, centimetres cubed per hour. This -1 per hour, this is literally meaning centimetres cubed divided by hours, because this is what the -1 bit means. If it was, yeah, so -1, it means divided by. So that kind of tells me that I am dividing a volume by the number of hours. So I kind of know how to get my volume. And this also reminds me, so here am I emphasizing these units, this also reminded me, literally as I was doing this question, oh my goodness, it's in 24 hours. So I have got to work out the volume and divide it by 24 to get centimetres cubed per hour. Now, already I'm kind of going, hold on, I've got centimetres here, but millimetres here. Stuff that for a game of soldiers. If they want centimetres cubed, I'm changing this into centimetres. 0.1 centimetres, okay? Because there are 10 millimetres in 1 centimetre. Now, that means that the radius is 0.05 centimetres. Now, just a little thing. Anything in a biology exam where they give you the dimensions of a tube of any sort, and I don't care whether it's a capillary tube or a blood vessel or whatever, they're always going to give you the diameter of the tube, never the radius, because thinking about it, nobody can measure the radius really in real life easily because you don't know where the center of the circle is. If you're trying to measure something, you can measure a diameter really easily. So here am I giving absolutely valid justification why they will never give you the radius. They will always give you the diameter. And you have got to just realize that 2 radii equal 1 diameter. All right, and I'm sure you do. So here is the equation, 3.14, squaring the radius times by the length, dividing the whole lot by 24. I'm then going to put that in standard form because all I'm working out is how many times I'm moving my decimal point, and it's 4 times. So it's 4.9 times 10 to the minus 4. There we go. So I got my 2 marks there. Hopefully that makes sense. Loving the fact you love respiration. I love respiration. Oh, do the 3 dots stand for therefore? Yes, this is therefore absolutely standard notation. As an examiner, I always accepted that for therefore. It was just, and this is slightly less conventional, but really very common. This upside down is because, and that's these 2 really useful for your revision notes. Don't use words, use symbols. There's my clue. Right, on to this one. Explain why log scale has been used to record the number of cells. Well, we're talking about flipping yeast cells. Oh my goodness. You know, yeast cells will go from 10 to 100 to 1,000 to, you know, I mean, they're doubling every generation. We are going to be going through several orders of magnitude. Order of magnitude is going from 10 to 100, i.e. going from 10 to the power of 1 to 10 to the power of 2, or from 100 to 1,000, 10 to the power of 2 to 10 to the power of 3. All right, and therefore that's why a log scale is used, because the number of cells range across several orders of magnitude. Didn't actually have to have that level of detail, just needed to know it was a large range. That would have been fine, would have got you the mark. Many cells die during the death phase. It suggests one reason why. Well, because they've run out of flipping food, I expect, because, you know, under laboratory conditions, you're assuming they're just sitting there in a pot. You know, new stuff isn't being added. So I would plump for using the word glucose rather than something vague like food or nutrients, personally. You are the biggest, I don't know what G means. Not to worry. Due to the lack of glucose. So there we go. That gives you the mark. Okay. Oh, or you could have put the yeast cells had produced too much, you know, something poisonous, because of course if they're anaerobically respiring, they'd be producing ethanol, alcohol, and carbon dioxide. So there we go. Okay, another bit of maths. This is deep breath maths. All right, just take a deep breath and go with it, because all you're doing is substituting numbers in to what looks like a flipping horrible equation. xt equals xo times e to the power of rt. Oh, flipping heck. You know, whatever. I'm just going to do as it tells me. Assuming no yeast cells died, yeah, okay, calculate the predicted size of the population after 10 hours. So this is X, so size of the population, so it is XT. I'm not having to rearrange it or anything. So XT, so X at 10 hours equals number at the start, 2,000, yeah, times by E, 2.72. Couldn't give a monkey's about this definition. Just give me the flipping number. And then to the power of R, growth rate, we're told it's 0.5 times T, which is 10 hours. So 2.72 times by itself 5 times by 2,000 gets you to this number. All of this I just put into a calculator. Clearly wasn't going to do it in my head. So there we go. If you got the right number, you got 2 marks, as always. Incorrect answer, but you got somewhere. You got 1 mark. Fair enough. Okay, on to be honest, as soon as I see graphs like this, this looks like succession. Yeah, where, you know, you kind of, okay, so investigating succession on sand dunes, they measured percentage cover, blur, fine. Okay, okay, here is a classic descriptor of a question that students do not know what it means. Describe how you would do something means write a method. It's not calculate it or do anything else with it. Write a method as to how you would do it. Okay, so how you would determine the mean percentage cover for beach grass, whatever beach grass is, on a sand dune. Okay, and here's the important other bit of reading the question. Mean. Do you know, I don't care if you've got no idea how to do this method. No idea. I don't care. If you've spotted, if you're being asked to describe how you would find a mean of anything, you can write 2 things. You can write, repeat the experiment many times, because obviously to calculate a mean, you need lots of numbers. And of course, if you're doing something biological, it's obviously many times. Always write many, don't write a number, and certainly don't write 3, write many. And also, I would also, if you've got no idea how to do the method, I'll tell them how you calculate the mean. You know, add up all the numbers, even if you wrote, add up all the numbers and divide by the number of numbers that you've got. Yeah, in this case, it is, you know what the numbers are, their percentage cover. Add up all the percentage covers and divide by the number of percentage covers that you've got. That would have earned you 2 marks if you'd just written that and you had no idea how to do the flipping method. So I know how to do the method. So here am I thinking that I'm writing it in exam speak. I know they often like you to say how you make that grid, you know, 2 tape measures at right angles. Don't have to write any more. They also like you to say how you produce how you place your quadrat randomly. Use a random number generator to produce coordinates. Place quadrat at coordinate site and estimate percentage cover of beach grass. Repeat many times. This is basically my method. I know it's going to have repeat many times in it. And then calculate mean percentage cover by adding up all the percentages and dividing by the number of quadrats, dividing by the number of percentages. Here we go. There you go. Look, I didn't even need this blurb at the front. Yeah, they just wanted you to know that they wanted the method of randomly placing the quadrats. So if you did nothing to explain that, you wouldn't have got that mark. Do it loads of times because it's a flipping mean, and say how you calculate the mean. Now, I'll be honest with you, I thought that this mark 0.3, I'm going to be honest, I thought that was a little bit GCSE. I wasn't sure that would get me the mark, but I scrabbled it in at the last minute because I thought that this first bit that I wrote was more likely to get me the mark than this. But anyway, it didn't. I got it that way. Okay. Oh my goodness, I have got no idea what you're talking about. O-level biology, would that be possible? Do you know, I'd love to, GCSE kind of obviously, but I've got enough trouble looking after A-level biologists and GCSE, yeah, hey, in the future, totally up for it. Okay, I just find A-level is the one that students really come unstuck on and really need the help with, and yeah, can make it nice and simple. Right, okay, they concluded that it was due to succession. Use figure 5 to explain why scientists read. So not use figure 5, use the flipping information you've been given. And this to me clearly shows succession, because it starts off with flipping beach grass being, being pretty much the only thing that is colonizing. And then a little bit later on, that dies, and whatever this is, scrub bunch grass takes over. And just as that's reaching its peak, hey ho, we've got a few trees, conifers and hardwood starting to add to. Notice this is mean percentage cover. Just take a note of what the y axis is. It's how much coverage of the ground there is. Okay, so yeah, just worth always noting. It isn't biomass. It's not number of plants. Always worth just kind of clocking what that y-axis is. Okay, so here am I about to launch into my description of succession using that graph to demonstrate that this is succession. So I'm thinking all my terminology. I'm thinking flipping pioneer species. I'm thinking, less hostile, abiotic factors changing. I'm thinking climax community. So here is my answer. Oh, didn't mean to do that. So species with the largest population, beach grass, is the pioneer species. This died and decomposed. I'd always write this, creating an increased soil depth. I have now named an abiotic factor that has changed as a result of a biotic factor, i.e. beach grass, being there, okay? And I was kind of thinking, okay, I've named an abiotic factor, and I've talked about it changing the abiotic conditions, which allows scrub bunch grass, SBG, to colonize and outcompete beach grass. Here am I thinking, I'm doing quite well. I'm using all my terms. Scrub bunch grass died and increased soil depth further. This increases, I risked using an up arrow for increased here. I was kind of thinking, oh crumbs, I'm on a time schedule. Yeah, I just risked this. This was extra. Increasing water retention and nutrients, and allowed conditions, wrote that twice, suitable for conifer trees and hardwood trees to colonize and outcompeting the scrub bunch grass. This became the climax community. Okay, so here's the mark scheme. So I got my mark point 1. I got mark point 2. When I saw this mark scheme, because really often mark point 2 and mark point 3 are actually all 1 mark in a lot of succession mark schemes. They either like you to name an abiotic factor that has changed, or they will allow you to say, oh, conditions became less hostile. I thought I'd covered that with this, my mark point 2 point. So, and so I was in, it's only because I happened to use the word suitable that I got mark point 3. I could have so easily missed that, so easily not got it. And I got mark point 4, as I would have expected, for naming the climax community. Okie dokie. All right. Thank you for the thank you. Hi, Carl. Would you consider do a bit? Weekend session. Oh my goodness. Oh, okay. All right. Josh, there's an idea. I'm not going to do it over the Easter weekend. I've got family stuff happening then. Okay. All right. Let me think. Let me think about that big weekend revision session. All right. The trouble is, it's always hard to know what are the hardest topics. Could put that. All right. Let me think about that, Josh. Let me think about that. Okay. Okay. All right. Let me have a ponder on that one. Easter revision session. I'm going to write it down. Okay. I don't know why the screen is always fuzzy, and then it takes a while to to clarify again, and I'm really sorry that it happens. All right, here we go. So we are on, also investigated how the proportion of sunlight reaching the ground changed during succession. Some of the results were obtained. Okay, fair enough. Use figure 5 to explain the results in figure 6. Here's the crucial thing. You've got to notice the difference in the x-axis. This one finishes at 500 years. This one, it's only just flipping started. So, and this makes sense because if all the trees are here at 500 years, doesn't it make sense that if we've got a high percentage cover, we are going to greatly reduce the percentage of sunlight reaching the ground? So, so here am I kind of going, use figure 5 to explain the results in figure 6. Well, the trees are reducing the amount of light reaching the ground. Okay, light intensity is reduced. Trees create shade. Wasn't quite sure what they're looking for. There we go. Trees block the sunlight. Notice they didn't want you to put blocks all sunlight. Okay, really easy to have said that. Right. Here, using evidence from figure 6, what can you conclude about the net primary productivity in the sand dunes that are older than 1,000 years? We're looking at this bit here. As soon as I see MPP or GPP, the first thing I do on the answer line is write the equation. So MPP equals GPP minus respiratory losses. GPP being gross primary production. Gross primary production is basically the amount of glucose that a plant can produce through photosynthesis. Some of that glucose is used up in respiration. The rest of that glucose can be used as the Lego molecular blocks, or the molecular Lego blocks, that make up all the other molecules that build the plant, that make up the plant's body. In other words, allow the plant to actually grow. So the glucose that isn't used in respiration can be used to make lipids, complex carbohydrates, proteins, DNA, all of those things that become the biomass of the plant. There we go. So MPP equals GPP minus R. And my basic thing is, well, if there's less photosynthesis, there's going to be less glucose made, and therefore there's going to be less body of the plant being made, less MPP. And so I got these two marks here. I thought mark points 3 and 4 were, sorry, just realized I didn't get mark point 4. I got mark point 3 saying that MPP decreased. Notice I didn't get mark point 4 because I didn't say that less light means less photosynthesis. I'd written it up here. So I thought that mark had gone, didn't write it, would never have written it. My equation didn't get me a mark, but for the length of time it took me to write it, I'd always write it as the first thing I wrote. Hazel, yeah, 5 o'clock every Tuesday, 5 p.m. every Tuesday. Okay, next question. Furosemide and CVT are drugs used to remove excess fluid from the body. They basically make you pee a lot. Okay, so to test this, they used 3 groups of volunteers, groups A, B, and C, gave them the same food. And then after 3 days, A got furosemide, B got CVT, and C got placebo, which means they didn't get either drug. And this is the mean volume of urine produced in 4 hours. God, I'm so pleased I wasn't part of this study. You wouldn't like doing this, would you? Pee in a beaker. Let's see how much urine you produce. Anyway, right. Let alone the smell. Anyway, moving on. Okay, all the volunteers were given the same food for 3 days. Suggest and explain. I'm noticing that both are required. Suggest and explain one reason why they were given the same food. So, I'm just kind of going for the obvious. My suggestion would be, is that food changes, the different types of food change the amount of the volume of urine you produce. Now, here's the thing. Notice that I am starting to quote what they say in the question. So notice I changed my wording to not amount of urine. I'm going, right, what was measured? Oh, it was volume. I'm going, right, it's going to change the volume of the urine. I'm going to use the wording they use in the question. Really useful for you not to stop you writing anything vague or in your own speak that you think is clear, but actually isn't exactly what they want to see. So worth noting that. So I would say that it would change the volume of urine. And then I was kind of going, not quite sure what to write after that. And so I kind of went for putting about the water potential of the blood, changing the water potential of the blood. I wasn't completely sure, didn't quite know what they were looking for, but it makes sense because the amount of urine that you produce is directly related to your blood's water potential. That is, ADH and all that kind of stuff. Oh, thank you for that, Hazel. I didn't want to know that. Guy who made artificial sweetener out of his, that will have been aspartame. Yeah, because oh, lovely. Yeah, I hadn't thought of that. Lovely. Manpreet, I'm hoping to. I'm going to be really honest, not quite sure when, but hopefully in the next few weeks. Hope that helps. So here we go. Here's mark point 1, mark point 2. Look, you can use this symbol for water potential. Always worth noting this if you see this in your exam board's mark schemes. Always notice abbreviations that they will allow. Okay, and they wanted volume. Also really notice that the word amount at A level rarely gets you a mark, rarely. You can nearly always replace the word amount for a more accurate description, whether that be, I don't know, volume, time, distance, area. You can nearly always replace it with a better word. It's a bit like your English teachers not allowing you to say nice. Right, using table 1, what can you conclude about the effectiveness of furosemide and CVT? Okay, right. Here's my advice to you. This is about reading the question. As soon as I read this question and noticed that they'd put in standard deviations, I absolutely knew that I was going to use that in an answer somewhere. It was going to be about standard deviations, either overlapping or not overlapping. And if in the course of the question, the whole question, I hadn't used that information, I would have known that I hadn't got a mark. I'd have lost a mark because I hadn't used that information. They don't put this information in for no reason. As soon as you see it, as soon as you see p-values, standard error bars on graphs, you absolutely know that you are going to be referring to them in a question, talking about them. So here it is. So I noticed that from this, that not only are both the drugs significantly better than placebo because the standard deviations don't overlap, also furosemide is significantly better than CVT because these standard deviations don't overlap. And I must admit, I felt like I was writing an awful lot for 2 marks. I thought this was a very meaty answer. And here's the mark scheme, and blow me down, they allowed you to talk about either the 2 drugs or the drugs more effective than placebo for 1 mark, which I thought was frankly stingy. And about the significant, because the standard deviations don't overlap. So that's that one. Oh, sorry, didn't mean to do that. Suggest how. Thank you, Cyber Ninja. Appreciate this, because this is going to be a long old thing, and I don't know whether I'm going to manage to do the whole lot. I have already made a video of this, which of course, and it will go up on the website probably in the next week or so. I've got to edit it. And that, of course, I'm kind of saying to you, you know, stop the video, read the question. But of course, I'm having to speak it all out because this has been happening in real time. Is sometimes treat high blood pressure. How would it cause a decrease? Well, it's going to be, if you're weeing a lot, you're removing water from your body, i.e. from the blood. Now, I would have just written, if I'm honest, I'd have just written this first sentence and would have been very close to leaving it at that. And I just, as an afterthought, I kind of went decreased blood volume equals decreased blood pressure. And to be honest, examiners may not recognize this as decrease. So be careful. This was me just shoving it in as a hopeful. And actually, I've actually given myself the mark here. But if I'm honest, I'm not sure I'd have got it, because I'm not sure a downward arrow is adequate for decrease. So that, hopefully that gives you an idea. If the standard deviations overlap, yes, it means that the mean, is not significant. The two means are not significantly different. You can't tell whether it's just chance or not. So hopefully that helps you. Furosemide inhibits the absorption of sodium and chloride ions from the filtrate produced in the nephrons. Explain how furosemide causes an increase in the volume of the urine. So here am I now applying all of that I know about how the nephron works, using the knowledge that furosemide stops sodium ions being actively transported out of, now let me get this right, what's the word? This is the ascending limb of the loop of Henle. So there was me kind of saying that, so sodium ions couldn't exit the ascending limb, therefore there would be less water leaving the descending limb by osmosis, and therefore there would also be less water leaving the collecting duct by osmosis. So that's kind of what I put. And here is the answer. I didn't put in the detail. I mean, to be honest, I was kind of thinking I'm running out of lines. I'm probably writing very big, to be honest, because I'm writing on a screen. So I got mark point 2 and mark point 3 because I talked about it coming from the collecting duct. It also does happen in the descending loop of Henle. Look, it allowed it here. Look, here we go. So just to kind of point out that my biology is correct. Okay, so I got 2 marks out of the 3. Frankly, happy with that. Bit of maths. Oh, this is, scientists measured the mean rate of flow of blood plasma. Notice it's blood plasma into the kidneys. Here we go. That's whatever, kind of going, whatever. I've got no idea what they're talking about. Okay. It's how much blood plasma goes into the kidneys. The mean rate of the flow of blood plasma is 60% of the mean rate of blood flow into the kidneys. Thanks for that. Okay, so this is 60% of blood flow. How much greater is the flow of blood into the kidneys with furosemide than with placebo? Over 4 hours. Give your answers in centimetres cubed. Here are your units, just a volume. So here am I here we go, I've underlined that. And here is my silly little diagram for me to get my head around the fact that these numbers are 60% of the number that I need to get to. I need to times this number for furosemide and this number for placebo by a multiplication factor that is going to give me the blood flow. And does it make sense that the fraction is 1 over 0.6? I could have equally have written 100 over 60 because this is 60% of the whole. So the fraction is 1 over 0.6 or 100 over 60 times by 380, which equals this. But then I notice it's over the 4 hours of the investigation. This is centimetres per minute. So I've now got to times this up by 4 hours times by 60 to get it for the number of minutes, which equals that. For placebo, this is the same calculation, which gives us that. This minus this is 34,000. One paltry flipping mark, I have to say. Stingy. Okay, so anyway, there we go. Moving on. 6. Right. Genetic crosses, observed phenotypic ratio are not the same as expected very often. 2 reasons, classic go-to reasons. Number 1, fertilization is random. Number 2, the sample size of the number of offspring is too small. You don't see a 3-to-1 ratio in a human family of 4 kids. Doesn't happen because the sample size, or very rarely, because the sample size is too flipping small. You know, humans have too few kids. Dear old Drosophila or peas, hundreds of offspring. You're far more likely to be closer to the ratio that you're expecting. Anyway, so there we go. Obviously, I got those 2 marks. There were others. I think these are far less obvious. They're reasonable. Okay. What is the genotype for this this individual? Well, it is homozygous recessive for both genes, and that was correct. Hopefully that is because it tells you big T is tall. Yeah, so it's just reading. It's reading the instructions in the blurb. Okay. Use the information provided to explain the results in table 3. Can I also just say the video that is going to be on the website is going to be completely clear. I'm really frustrated that this is just looking so blurred on YouTube, and I honestly don't know why. I hope this is still useful. I'm going to keep going for a little while at least. Use the information provided to explain the results in table 3. Well, they have told you in this, they have told you that they've told you the numbers for, you know, what I would call the double dominant phenotype and the double recessive phenotype. I'm looking at this and going, this is a 3-to-1 ratio. And also they show you the position of the dominant and recessive alleles. They are clearly on the same chromosome. So use the information to explain the results. Well, this is autosomal linkage. But the question is why on earth did these, what I very technically call the in between the mixy-uppy phenotypes, why on earth did we get a stupid, silly small number of each of these? Well, that was because crossing over occurred. So you actually ended up with a few gametes that were little t and big M and big T, little m. So 3-to-1 ratio of tall, normal to dwarf mottled plants shows that the 2 genes are autosomally linked and crossing over has occurred to produce the other 2 phenotypes. And I then went on to say that you would only expect to have big T, big M and little t, little m gametes, whereas there were a small number of these gametes produced. And if you look at the mark scheme and the notes, I picked up mark point 1 saying it was autosomally linked, and I picked up mark point 3 because crossing over. Didn't look like I'd picked up mark point 2, and I was feeling very hard done by until I looked at the notes here, which I am allowed, I got the mark with that bit there. Completing table 4, well, we put in all the phenotypes, and this is if they're on separate chromosomes, put in all the phenotypes. This would be your classic 9:3 to 3:1 ratio. And the way to know which phenotype has which ratio is that the 9 is always what I call the double dominant phenotype. The 1 is the double recessive one, and the 3s in the middle are the mixy-uppy ones. So, and I got both columns right, so I got those answers right. Oh, good, quality's fine, good. On maybe it's just because my second screen is my iPad. That's how I'm reading all of your chat. And it's probably just because it's too small. Sorry, I'm being a bit thick, obviously. Okay, question 7. Let's just see if we can get through this. Dopamine, neurotransmitter released in synapses in the brain, similar to that of acetylcholine. You learn about cholinergic receptors. Dopamine stimulates the production of nerve impulses. Okay, so it basically does the same as acetylcholine. Describe how. Do not include events leading to the release of dopamine and events following the production of the nerve impulse. Okay, so I'm going to make sure I only do what they ask. So my question, I've deliberately started it once released. So we've got the dopamine released from the presynaptic neuron. That dopamine diffuses across the synaptic cleft, binds with receptors on the sodium ion channels, on the postsynaptic neuron, and this causes sodium ions to diffuse into the postsynaptic neuron, depolarizing the membrane and causing the initiation of an action potential. And I'm thinking, I've nailed it. Dopamine diffuses across, yeah, fine, got that. They didn't even require a synaptic cleft. I didn't get mark point 2. I did get mark point 1. Yeah, I did get mark point 1. I didn't get mark point 2 because I didn't talk about this postsynaptic membrane. At which point I kind of go, oh, do you know, whatever. I couldn't really have written much more and made it much clearer. This is a classic case of, you know, forget it, have the mark. I never would have written anything different, if I'm honest. Do I try and make note of they want to know postsynaptic membrane rather than neuron? I'm kind of confident, if I'm honest, I'm kind of sitting here as an A-star candidate. This is me, it sounds really big-headed, but clearly I know my biology. And I'm kind of going, I haven't picked up every mark on this exam. Actually, I'm not going to worry about it because I'm really confident that I'm picking up enough to get the grade I want. And that's the crucial thing. What you want to be doing when you're looking at mark schemes is noting those mark points where you kind of know, oh, I really should have got that, either because I didn't read the question carefully or I knew that and it would have been an easy way for me to get a mark. But let's be honest, if you're some way confident that you're in the right range of marks to be getting your grade that you want, there are some marks that you just kind of go, oh, whatever. That's a whatever mark for me, rightly or wrongly. Right, here we go. Morphine. Release of dopamine stimulated by chemicals called, dopamine is stimulated by chemicals called endorphins. All right. Endorphins attach to opioid receptors on the presynaptic neuron that release dopamine. So this is how endorphins stimulate the release of, this is how endorphins stimulate the release of dopamine. They attach to the opioid receptors, bringing about the release. Morphine is a drug that is similar structure to endorphins and can provide pain relief. Here we go, look. Dopamine includes pain relief. So morphine, now notice in my answer that I am using the terms that they have given me in the question as terminology in my answer. Morphine binds to opioid receptors on the presynaptic membrane, which brings about the release of dopamine. Here we go. And therefore, I've got the 2 marks as a result. They absolutely wanted you not to just write a standard answer. Oh, it binds to receptors. You know, they've told you stuff. Use what they've told you. GABA, neurotransmitter released by inhibitory synapses in the brain. Inhibitory synapses are ones that tend to stop the nerve impulse via neurotransmitters jumping across the synapse and continuing an action potential. They tend to stop that happening. They inhibit. GABA, and they actually GABA causes negatively charged chloride ions to enter postsynaptic neurons. That makes sense to me. Explain how this inhibits postsynaptic neurons. So remember, in the postsynaptic neuron, just as it will be along all the rest of the neuron, there is more positive charge on the outside than there is on the inside, making the inside, in comparison to the outside, approximately minus 70 millivolts, that resting potential. Notice that the minus 70 is the inside in comparison to the outside. And this question tells you that GABA causes negatively charged chloride ions to go into the neuron. Well, if we're adding more negative charge to already the negative part, it's going to cause the negativity of the inside of the membrane to drop even lower. So it won't be minus 70, it might be minus 80, minus 90. It's going to get even more negative. And there's a word for that. It's called hyperpolarization. Remember from your action potential graph. So I've used that word because I know what it means. The chloride ions entering postsynaptic neuron causes hyperpolarization. And I'm kind of going, that's my headline answer. I think I've got 1 mark. All right. So what else am I going to write? How does it inhibit? Oh, okay. If it's really negative, that's going to mean that more sodium ions have to diffuse into that membrane in order to reach that threshold so that an action potential is kicked off, that all or nothing response. And therefore, that's what I've written. Oh my goodness, Mohammed, I would love to do CIE. I've just got a big enough job to do covering the main exam boards for UK. Can I just say to you though, that the degree of the detail and the degree of terminology, I know it's not identical between exam boards, I really know that, but the kind of terminology and me talking about certain topics are going to help you enormously in understanding those topics. So I'm hoping that this is at least useful, giving you an idea as to the kind of level of detail that you need to know and the kind of terminology that you need to use in exam answers. So here's the mark scheme. So I've got hyperpolarization, more sodium ions required for action potential, got all 3 marks. Okay, 8, what is meant by genome? Genome, right, I'm going to be really honest with you. This is what I wrote. All the genes in an organism. And I suddenly kind of went, oh my goodness, I've just written that as a knee-jerk response to seeing the word genome. And I went, it's not all the genes, it's all the DNA. It's not just the genes, it's everything. So I kind of thought, crumbs, just, you know, dodged a bullet there kind of thing. That would have made me look a bit stupid if I hadn't got that right. All the DNA in a cell, fine. And then I looked at the notes, except all the genes. Couldn't believe it. I was really surprised by that. Anyway, okay, so I got the mark, done that. Okay. Explain why the antibody, so now we've got to read this blurb in order. So this, I can't even remember what the context of this is. Chromatin immunoprecipitation is one method to determine where a transcription factor binds to DNA. All right, whoopee-doo. Right, fine. Okay, go with it. Add chemicals to bind to the transcription. To bind the transcription factor to the DNA. Break down the DNA into small fragments, add an antibody to the transcription factor. So we've now got an antibody on the transcription factor on the DNA. Remove the precipitated DNA, add chemicals to remove the antibody with attached transcription factor. Removing the transcription factor and the antibody from the DNA. Amplify the DNA, which will correspond to the site occupied by the transcription factor. Okay, you know, whatever. Right. Explain why the antibody binds to the transcription factor. Well, that's because they're complementary. All right, so here's what I write. Antibody has a variable region, it's called the antigen binding site, which is complementary to the shape of the transcription factor. So we have got complementary shape. Right, I gave myself, and I looked at the notes, I gave myself mark point 2 for complementary. I didn't give myself mark point 1 because I thought that this word is important, that it has a specific tertiary structure or shape. So I either had to use the word specific or I had to use the word tertiary structure. Tertiary shape? You'd never say tertiary shape. I didn't use the word specific, and again, look, here has a specific variable region. I didn't use the word. And I think that if I had written, which is complementary only to the shape of the transcription factor, then I would have got mark point 1. Because using that word only, can you see, means exactly the same as saying it's specific. It only binds to one thing. That is the definition of specific. So, you know, I didn't get it. So not terribly worried. Okay, what precipitated DNA consists of? Well, I kind of went through my logic. The precipitated DNA is everything all added together, transcription factors, antibody, and DNA. Here is the answer, so I got the mark. Right. Soybeans, using a number of processed food, they contain a protein known as P34, which causes an allergic response. Okay, so we don't want P34. So they've created a transgenic soybean that produces single-stranded complementary DNA, which prevents transcription of the P34 gene. All right, okay, weird, but true, must be. Can I just say, all of the questions are based on research. They are true. All right, so they don't make the questions up in that sense. They use proper science. They used recombinant plasmid as vectors to transform the. So okay, after they had screened these cells for production of the P34 protein, they cultured. Right, okay. Suggest how single-stranded cDNA could prevent the transcription of the P34 gene. Well, I'm kind of thinking, okay, single-stranded complementary DNA. If it's single-stranded, that means it could bind by complementary base pairing to the messenger RNA, because messenger RNA is single-stranded, yeah? Can't bind to the DNA. DNA is double-stranded. The DNA of the soybean can't, it's got to bind to the mRNA, the way I'm thinking. Two single-stranded nucleic acids, they can bind to each other if they've got complementary base pairs. So single-stranded cDNA binds to mRNA of P34 genes, means that the ribosome can't bind to the mRNA to do translation. So, and I got that first mark point here. They allowed you saying that it bounded to the DNA. I don't, anyway, fine, that, fine. Or they allowed you to say that. That wasn't as obvious to me, so I got the mark. Describe the roles of two named types of enzymes used to insert DNA fragments into plasmids. So the classic ones, in my view, are restriction endonucleases, which you've got to use to cut out the DNA fragment and the vector so that the ends can join together, and DNA ligase, which binds the DNA fragment into the plasmid by forming the phosphodiester bonds. And as it was, I wrote too much, but I got both the marks. Okay, oh, sorry, didn't mean to put that one up. Soybeans processed right, they were screened, involved gel electrophoresis to separate the proteins. Well, gel electrophoresis separates both proteins and DNA on the basis of two things, on their length or their mass and their charge. So I put different lengths, different charge. I got mark point 2 and I'm thinking, no, don't tell me I haven't got mark point 1. Except wait for mass. Oh, flip it. Thank goodness. Right. Notice they would not have allowed the word size. Be more precise, particularly this whole gel electrophoresis things. You can't just say, oh, it's down to the size of the DNA or the size of the protein. Be more specific. Number of amino acids, number of nucleotides, length, mass. Yeah. Yeah, I wouldn't have thought of writing R groups as mark point 3, so I've got those 2 marks. Okay, we're on to 9, we're on the home straight. Okay, right. Lactose is the main sugar in milk and is hydrolyzed by the enzyme lactase. Here am I writing little notes as I'm reading this. So lactase forms glucose and galactose. I'm immediately thinking that. Here's lactase, that's what speeds it up. If this happens in young animals, most young mammals, absolutely, because obviously young mammals drink milk, when they're babies. They stop producing, generally they stop producing lactase as they grow up, but humans are an exception. They produce it as adults, and we call this lactase persistence. In other words, the lactase is persistently being made, and it's controlled by a dominant allele. Okay, selection pressures. Okay. One hypothesis suggests that the selective pressure was related to some human populations farming cattle as a processor. Okay, describe how farming cattle as a source of milk could have led to an increase in lactase persistence. Well, here we're talking about evolution and natural selection, because we're talking about a change in the allele frequency. And a change in the allele frequency is the definition of evolution. So, and I think, doesn't it say selective pressure? So we're talking natural selection. So here is my answer. Here am I applying my knowledge of natural selection to the scenario of the question. The allele for lactose persistence occurred because of mutation. First thing, if I'd been describing natural selection, the first thing I would have written is there is variation in the population. Yeah, that variation comes about as a result of mutation. Didn't need the first bit. This would be an advantageous allele because being able to digest milk in the populations farming cattle would increase those individuals' chances to survive and reproduce. That is a flipping long sentence. I could have done with splitting it into 2 sentences. Nearly always shorter sentences make your answer clearer. That's quite a long sentence. This passes the LP allele down to the next generation, increasing the LP allele frequency. I've got to the end of the question. How farming cattle as a source of milk, I've talked about being able to digest it. I'm thinking that's a pretty reasonable answer. So I got mark point 1. Okay, oh, and here are the notes. I got mark point 1. I didn't get mark point 2. Which is really annoying because I'd flipping written it up here, realizing that lactose is glucose and galactose. And I talked about individuals with LP more likely to, sorry, I did 5 frequency. Yes, I've done that. Oh, sorry. I've also got 3. Got 3 here. So I got 3 out of the 4. I missed directional selection. I missed the named nutrient, which, okay. Why does the number of people showing LP rapidly increase? Well, that's because the allele is dominant and it is shown in both homozygous dominant and heterozygous individuals. So I got both the mark points there. So here they tell you that the mutation does not occur in the gene. So you have now got to think about somewhere else in the DNA where this mutation could have occurred that would affect the transcription of the protein, of the lactase. So my immediate go-to would be transcription factors. So I've just said mutation in the transcription factor for lactase gene. The transcription factor now binds to the promoter site. So the mutated transcription factor now binds to the promoter site of the lactase, causing the lactase gene to be expressed. Here's the mark scheme, and I got mark 0.1 here, that one, and causing it to be transcribed too. Right. Oh my goodness, I'm exhausted. I don't know about you. Question 10, last question. This is the comprehension question. I'm not going to read through the whole thing. It's all about rods and cones and problems with rods and cones, and some of them being recessive issues and some of them being sex-linked recessive issues. I'm going to copy and paste the appropriate part of this blurb for the sentences, if that makes sense. So people with complete achromatopsia, yes, have difficulty in seeing detail. Here we go. Complete achromatopsia is a form of complete color blindness. It is caused by only having rods and no functional cone cells. They have difficulty in seeing detail. Explain why. Okay, right. This is because people with complete achromatopsia have no functioning cone cells. Notice I'm using the terminology in the flipping question. I'm not saying they have no cone cells, because that isn't true. They have no functioning cone cells. And that means that if their cone cells don't function, if you remember, the cone cells are connected to one bipolar neuron for each cone cell. This is different for rods. Rods have many rods connected to one bipolar neuron. Because cones are connected to, for each cone, they're connected to their own bipolar neuron, it means that if 2 points of light hit 2 cones, the brain receives 2 nerve impulses and therefore perceives 2 points of light. That doesn't happen with rods. If the 2 points of light hit here and here on the 2 rods, because they are connected to only one neuron, the brain only gets 1 nerve impulse. It only perceives 1 point of light. That's why cones give you much greater acuity. They increase your ability to see detail. Oh, I've just realized I've summarized it here. I've forgotten that. Anyway, okay, I'm gonna move on. So here is my answer. Because their cones are non-functional. Cones are connected to a single bipolar neuron. This means that 2 points of light detected by 2 cone cells will initiate, I can't read it underneath, send 2 impulses to the brain, therefore 2 points of light. So I got mark points 1, 2, I thought I got 3. Yes, I did get 3. Okay. 10%, they tell you down here we go, 10% of the population on this specific island of Pingelap, are affected by complete achromatopsia. Use the Hardy-Weinberg equation to calculate the percentage of this population who are heterozygous for this disorder. Notice that they are giving you phenotypic information. The 10% of the population are displaying the characteristic of achromatopsia. And you have got to come up with the percentage of the population. This is what they're giving you. There it is. This is what they've given you, and this is what you've got to give the answer as. population that is heterozygous for this disorder. Now, those that show complete achromatopsia, it's an autosomal recessive allele. So I'm kind of going, hey, it's little a, and the proportion is 10%. 0.1 equals q squared. Now, this is because the Hardy-Weinberg equation, p squared plus 2pq plus q squared equals 1, is talking about the genotype of the population and therefore the phenotypes. Q squared being if you remember, the homozygous recessive genotype, q times q squared. So 0.1 equals q squared. Notice that it equals q squared, not q. So therefore q is 0.32, square root of 0.1. Because the other Hardy-Weinberg equation, p plus q equals 1, this means that q is 0.68. We can now work out the proportion of heterozygotes using 2p squared. 2 times 0.32 times 0.68. Now that will only give us the proportion of the population, not the percentage. We have to times that by 100 to get the percentage, 43.52, which is right. Okay. Colour blindness affects men more than women. So here we are, sex-related recessive allele. That's because this is, so it just tells you it's sex-linked. Sex-linked means that the gene is only on the X chromosome. And certainly at A level, there are no alleles on the Y chromosome. It's only on the X chromosome as far as A level biologists are concerned. So if it's only on the X chromosome, that means boys or males being XY chromosomally only have the possibility of carrying one allele or having one allele. If they inherit the recessive allele, they will be red-green colorblind. They will have that characteristic. However, women who are, or females who are XX, have to have inherited 2 recessive alleles. XA, as it were. Oh, sorry, XR, whatever, it doesn't really matter. You know, I have to have inherited 2 in order to be colorblind. So therefore, just by chance, it's far more likely that the condition is going to be shown in men than it will be in women. Oliver, hi. I'm in year 9. Should you be watching this? No, this is far too complicated for you. This is A level. I'm loving the fact you're keen, but no, go and have a cup of tea. Okay, all right, so I got both the marks there. I did have to run over the lines, but I think it's because I'm writing big because I'm on a screen. Final questions. People with red-green colourblind are unable to distinguish between red and green and also between other colours. Here we go, also between other colours. Well, that is because cones are the cells that detect light, and we have 3 different types. We have red cones, blue cones, and green cones in our eyes, each of which are sensitive to red light, blue light, and green light. And it's because you have to have all of those cones functioning for you to be able to see all shades of colour. So what did I put? Because they have no non-functioning green cone cells. We have 3 different cone cells detecting red, green, blue light, and all 3 need to be functioning to distinguish between all colours. I fell down here. I didn't get mark point 1 because I didn't talk about green sensitive cone cells. Notice I didn't talk about, where is it? Green sensitive cones. I didn't use the phrase in the flipping question. Look, reject reference to green cones, blue cones, and red cones because the cones in your eyes haven't got different colours themselves. They detect red light, detect blue light, detect green light. So I got it right here. I made it clear here. We have different cone cells detecting red, green, and blue light, but I got it wrong here. So I didn't get mark point 1 once only, but I only got it wrong once. I talked about different cone cells. I got that. And I wasn't convinced that I'd written enough to get mark point 3. So I didn't give myself the mark. I just felt this was a little bit vague. They needed to be quite frank, I was getting tired. Right, last one. Sorry, let's not do that. Last one. Current research into the treatment of red-green colour blindness involves the use of induced pluripotent stem cells. Whee! Suggest how induced pluripotent stem cells could correct red-green colour blindness. Well, here am I. As soon as we talk about stem cells, I'm thinking about the definition of stem cells. 2 parts of the definition. They replicate cells, and they can differentiate into other cells. And induced pluripotent stem cells can differentiate into practically every other cell. Fantastic. So I wrote, most obvious thing to me seemed to be to say that they can differentiate into, I got it right here, green sensitive cones, and they can also divide continuously to replace themselves so that it could be a long-term treatment. Oh, I went beyond myself here. So I got the 2 marks. Okay, got the 2 marks. This is the final question. This is it. The use of IPS could have advantages over the use of gene therapy to correct red-green colour blindness. Use the information from the passage, suggest and explain reasons why. So here am I kind of going, oh my good, oh, do you know, I'm absolutely, by this point, I've realised that what I've written in the previous answer is absolutely the answer to this question. Yeah. So really obvious, number 1, that iPS cells can replicate indefinitely and therefore provide a long-term treatment. Thought that was reasonable. Also, I then went and looked and kind of went, okay, what kind of going, okay, I've got 1 out of 3. Suggest and explain. So suggest they can be long-term, and explain because they divide. Then I'm kind of going, okay, what's the rest? Okay, maybe they're better than viruses because, so better than viruses, there's my suggestion. Explain because viruses could mutate and become pathogenic. And now I was really feeling like I was dredging the bottom of the barrel. I'm kind of going, mark point 3. And then I had a mini revelation that induced pluripotent stem cells have, of course, come from adult cells. So if my immediate thought, oh, well, if they took an adult cell from the patient and then made induced pluripotent stem cells and injected them into the person's eye, then that would mean that there wasn't any rejection. Apart from the injection itself. Anyway, moving on. Okay, so, and therefore, this is what I wrote. Felt like I'd got loads of lines, felt like I was able to write exactly what I wanted to. Here is the mark scheme. And so I got mark point 1, mark point 2, and mark point 3. And there finishes the flipping exam. Paper 2, June 2018, AQA. I really hope that was useful to the number of you that are still watching. Well done, hanging in there. And as I said, a much neater and more efficient version will be up on my website in about a week's time. In the meantime, I am going off to have tea. I can smell it's being cooked for me. Looking forward to it. And I will see you next week. Doing whatever is voted on my community tab in this next week, either going through an OCR exam paper, doing some part of biotechnology, or what was the other thing? Doing something about conservation. Anyway, cast your vote. See you next Tuesday at 5 o'clock. Bye-bye.